The mouse body is covered with hair and is divided into three parts: head, trunk, and tail. On the head are located the mouth and the sensory organs, external nostrils, eyes, ears, and whiskers. The triangular mouth is ventral and is surrounded by two lips, under which we can see two pairs of elongated incisors, which have continuous growth, which is typical of a rodent. Eyes are protected by two eyelids. The iris of the white mouse has no pigments, and its red color is due to deep blood vessels. Two skin folds, which are outer ears or auricles, surround external ear canals. Their base is strengthened by a cartilage scaffold. There are tactile hairs or whiskers on the head. Like all mammals, whiskers are distributed into four groups, divided around the mouth, on the throat, and above the eyes. The trunk has a pair of limbs on each side. The mouse has pentadactyl chiridium limbs, and these are divided into three segments. The forelimb is composed of the arm, the forearm, and the hand. The hind limb is composed of the thigh, the leg, and the foot. The hand has 5 fingers, of which only 4 are developed and have claws. The foot has 5 well-developed fingers ending with claws. The whole foot comes into contact with the ground. The mouse is a plantigrade animal. Hands and feet have pads on the fingertips and palmar and plantar calloses. On the ventral side of the female trunk, there are 5 pairs of teats, divided into 2 groups, which extend over the pectoral and the pelvic region. The male's teats are atrophied and not clearly visible. At the base of the trunk, in the ventral position, we find openings for the alimentary posterior canal and the urinary and genital systems. The anal orifice is visible from behind for both sexes. For the male, a cutaneous pouch, the scrotum, hides this orifice. The position of the urinary and genital orifices differentiates male and female. For the female, the vagina is found anterior the anus, at the vulva area. A perineum separates the anus from the vulva. The urethra opens anterior to the vagina at the extremity of the urinary papilla. So the urinary and the genital tracts are utterly separated in the female. In the male, genital and urinary orifices merge at the extremity of the penis located in a skin fold, the foreskin or prepuce. The prepuce orifice allows the projection of the penis in erection for mating. A large perineum, covered by the scrotum, separates the anus from the foreskin orifice. The tail is as long as the rest of the body. It's covered with horny epidermal scales arranged in rings, between which there are short hairs. In order to observe the organs in place, the mouse must be opened on the ventral side. The mouse has to be laid down on its back. Then, stretch the limbs perpendicularly to the body axis and fix them with pins in the hands and feet. It's necessary to locate the prepuce orifice in the male and the urinary orifice in the female. Make an incision in the skin with fine scissors about a couple of millimeters above the orifice. From this opening, the incision must continue on the mid-ventral side up to the chin. Two lateral incisions extend towards the extremities of forelimbs and hind limbs. The skin is separated from the underlying muscular layer and pinned on the sides. In this step of the dissection, at the base of the head, we can notice a part of the salivary glands. Next to the salivary glands, the main muscles of mastication, digastric and masseter muscles, allow the mouth to open and close. At the trunk level, we distinguish two regions, the thorax and the abdomen. The abdominal cavity must be opened to highlight the organs in place. Abdominal muscles must be lifted and incised up to the base of the thorax. Then, two transversal incisions are made at this level to complete the abdominal cavity opening. Muscular foldings are pinned on the sides. The liver and a part of the intestines are now visible. In its upper part, the abdominal cavity is separated from the thoracic cavity by a very well-developed muscle, the diaphragm. In the ventral part, the diaphragm follows the costal arch. The rib cage is dorsally limited by the spine, sideways by the ribs, and ventrally by the sternum. The thorax dissection allows to observe organs that are in the thoracic cavity. A grooved director is inserted under the sternum and slipped towards the head and exits at the neck level. The sternum is cut with scissors, following the grooved director. Both halves of the rib cage, now freed, are drawn aside by the action of the pectoral muscles. The diaphragm is cut at its periphery, following the costal arch, and the rib foldings are removed. The lungs, the heart, and the thymus gland are now visible. The thymus is an immune organ involved in T cells maturation. The respiratory system includes the larynx, the trachea, and the lungs. To better see the larynx and the trachea, the salivary glands and the neck muscles are removed. Both of the thymus lobes are excised. Now we can see the larynx and the trachea, strengthened by a set of cartilaginous rings. The right lung is divided into 4 lobes. The left lung is made of only one large lobe. At the base of the larynx can be seen the thyroid, an endocrine gland which is not part of the respiratory system. During breathing, air comes in through the mouth and nostrils, goes through the pharynx, the larynx, and enters the trachea. Then it follows subdivisions of bronchus and bronchioles to arrive in the pulmonary alveoli, where the gaseous exchange will happen. Alveoli are surrounded by venous and arterial capillaries that transport blood. The exhaled air will take the opposite path. In mammals, breathing is done through the volume variation of the rib cage. During inspiration, the rib cage expands by contraction of the intercostal muscles and the diaphragm. Air pressure in the lungs is reduced to below atmospheric pressure, and air enters the lungs. Exhalation happens when intercostal muscles and the diaphragm relax. Thus, the thoracic cavity returns to its initial volume. The mouse circulatory system is closed and divided into two parts: the pulmonary circulation between the heart and lungs, and the systemic circulation between the heart and the other organs of the animal. The heart is completely partitioned and is divided into a right and a left heart. Each part includes an auricle and a ventricle. The left ventricle is more developed than the right one. Blood circulation is ensured by alternated contractions of auricles and ventricles. Carbon dioxide-rich blood, coming from the head and from other parts of the body, join the venae cavae that enters the right auricle. The auricle contraction allows blood to go through the right ventricle. Blood leaves this ventricle, passing through pulmonary arteries, and flows to the lungs for oxygenation. Oxygen-rich blood leaves the lungs, passing through pulmonary veins, and returns to the heart in the left auricle. Then it passes through the left ventricle, which sends it into the aortic arch. The latter extends into the aorta, from which arteries branch towards the head, the forelimbs, and the rest of the body. The mouse's digestive system is composed of a complete digestive tract, going from the mouth to the anus, and by additional glands. The digestive tract is divided into four regions: the esophagus, the stomach, the small intestine, and the large intestine. The esophagus is a simple tube going from the mouth, through the neck, the thoracic region and the diaphragm, to the stomach. To better see the esophagus, it's necessary to move the cardiorespiratory system that partially covers it. To do so, blood vessels have to be cut. The vena cava is cut at the diaphragm level. The aortic arch is severed. Followed by other vessels and connective tissues attached to the trachea and to the cardiorespiratory system. These tissues must be removed with the help of forceps going up along the esophagus while holding the heart and lungs apart on the left. The heart and lungs are pinned. The esophagus is now visible in its whole length. It meets the stomach in the abdominal portion. The third part of the digestive tract, the intestine, is limited to the abdominal region and is divided into the small intestine and the large intestine. To observe these regions, the digestive tract has to be unrolled by going gradually back from the rectum up to the stomach. Mesenteries that link the intestinal folds are delicately torn by pulling on the digestive tract. The unrolled intestine is pinned on the side of the animal. The small intestine is divided into three parts: the duodenum, the most anterior, the median jejunum, and the posterior ileum. The large intestine is the shortest and is composed of three parts as well. It begins with a dead-end pocket, the caecum. It continues with the colon and ends with the rectum leading to the anus. The rectum can be easily recognized thanks to the presence of a string of droppings inside. The digestive tract also has additional glands, the salivary glands, visible in the neck part, the liver, and the pancreas. The liver is divided into 4 lobes. The gallbladder is associated to the liver. To expose it, lift and move aside the median lobes of the liver. The common bile duct carries all the hepatic secretions to the duodenum. The second additional gland, the pancreas, has a plumose shape and is located behind the stomach. In the abdominal cavity, the spleen is also present. It is an immunological organ which is not part of the digestive system. The observation of the urinary system, which is located in the abdominal cavity, requires the dissection of the alimentary canal. This urinary system is composed of 2 kidneys, which produce urine that's excreted through the ureters, the bladder, and the urethra. In both sexes, the urinary system is identical up to the bladder. The kidneys are 2 brown-colored bean-shaped organs located on the dorsal side of the abdominal cavity. On the kidney's anterior region, we can see the adrenal glands, surrounded by adipose tissue. These 2 endocrine glands are not part of the excretory system. The ureters, 2 canals that transport the urine to the urinary bladder, begin in the concave part of the kidneys. The bladder is a spherical bag located behind the abdominal wall, which is used to temporarily store urine. Urine is eliminated through the urethra, a canal which has only a urinary function in the female. In the male, the urethra has a urogenital function. It winds through the center of the penis and also allows the emission of sperm. The pelvic symphysis opening allows us to observe the urethra. The symphysis, located under the urinary bladder, corresponds to the assembly of the two-half pelvis at the level of the pubis. Once the pelvic symphysis has been severed with the use of scissors, both halves of the pelvis are moved aside. This dissection allows to observe the urethra path. That ends at the level of the urinary papilla in the female. In both sexes, in order to observe the reproductive system, located in the abdominal cavity, requires to unroll the intestine and the pelvic symphysis section. The female reproductive system is made of two gonads, the ovaries, two oviducts, the uterus, the vagina, and additional glands. The ovaries are two small, ovoid, pink-coloured organs covered with adipose tissue and located beyond the posterior edge of the kidneys. Facing the ovary, the short oviduct starts in an auricle that collects oocytes. The oviducts join the uterus in the shape of two horns which merge into the uterine body. The basal region of the uterus, the cervix, joins the vaginal canal that opens on the outside at the level of the vulva. The uterus and the vagina are partly hidden by the bladder and the urethra of the urinary system. At the base of the vagina, two clitoral glands can be noticed, and they lead directly to the outside. The male reproductive system is made up of two gonads, or testicles, two vasa deferentia, the penis, some muscles, and a series of additional glands. In the mouse, the testicles are two ovoid structures found within the abdomen. They move down into a cutaneous pouch, the scrotum, during mating and after the animal's death. Remove them from the scrotum by pulling on the whitish adipose tissue that covers them so that we can observe them. Fold and fix the scrotum sections, and then pull and pin the adipose bulks of the testicles by the sides. Suspensory ligaments can help hold the testicles in place in the abdominal cavity. Circuitous canals, the epididymis, extend from each testicle and move spermatozoa into the vas deferens, or ductus deferens. The observation of the reproductive system as a whole requires the opening of the pubic symphysis. The vasa deferentia appear at the base of the urethra, which is located in the middle of the penis. In the male, this canal is common to the urinary and the reproductive system. Two muscular masses can be seen at the base of the penis. The ischiocavernous muscles, that contribute to erection, and the bulbocavernous muscles, which play a role in the emission of urine. The male reproductive system includes additional glands which take part in the production of sperm: the seminal vesicles, the prostates, and the Cowper's glands. Ram's horn-shaped seminal vesicles are located at the base of the bladder. The prostate comprises 3 pairs of accessory glands necessary for seminal fluid coagulation. The most developed pair, the coagulating gland, is located within the seminal vesicles concavity. The two other pairs, the ventral and dorsal prostates, are visible at the base of the bladder. The section of ischiocavernous muscles allows the observation of the Cowper's glands, which produce secretions important during mating. The Tyson's glands are accessory glands located between the abdominal muscles and the skin. These cutaneous glands do not take part in the production of sperm and correspond to the clitoral glands in the female. The Tyson's glands secrete pheromones and lubricating compounds for mating.
A bit.
There you go. Whoa.
You can see the foramen magnum. You can see it on the screen too. It's that white spot. That's basically that hole in the skull that you learned during anatomy. That's where the spinal cord leaves from. I'm going to use that as my target, and I'm going to actually use the little scissors and cut up and around almost like a can opener. I know that sounds gross, but it's not meant to. It's an actual analogy. You can hear the skull bones breaking there pretty good, right?
Wow.
So I'm just basically taking this on each side.
Brain surgery.
You don't have to do that, no. That's why I'm doing this here, in case you don't want to do it. But if you do this, what you want to do is get a probe or something underneath there so that you can lift up, almost like a pop tab, on the back part of the skull. I'm just pushing the brain down to make sure it doesn't pop up. If you do it properly, stay down. Just keeping the brain down. And pop in the pop tab.
Whoa.
If you do that, you'll start to reveal the brain. Just continue this cut. Is it still in there, Mike?
Yep.
Okay.
That's awesome.
Actually, I should have had Mike do this. He's senior research has involved a lot of brain isolation. So there's obviously some really nice pictures because I'm a neuroscientist. I do this a lot, and so I have some really nice pictures in your rat dissection guide that you'll see. But there's nothing like seeing it, the real deal, like in person. Yeah, that rat had a thick skull. So anyway, so that you can actually fold out of the way gently and like as.
Move it up a little bit.
Oh, okay.
Perfect.
Allie, you still good there? You sure? I'm going to use the other side of the scalpel just because it's a flat surface so that I can take this thing out. Just kind of roll it out so that you can see the underside as well as the, oops.
Oh,
we lost it.
So
After sacrificing the mouse by cervical dislocation and extracting the brain from the skull, rinse it in cold PBS to remove excess blood. Then place the brain on the ventral side facing the cold plate. To prevent the brain from sliding too much, you may add some blotting paper moistened with PBS on the cold dissection surface beforehand. Place a scalpel blade perpendicular to the plate and cut through the entire midline of the brain, following the groove of the interhemispheric fissure. Then gently pull apart the two brain halves. You can then dissect each half in parallel, or you can do one after the other. Here I will dissect the left half first and then go on to the right side. Place the half brain with its lateral side facing up. Since this is fresh tissue, you can easily distinguish the different parts of the brain as they differ in colour. Using the scalpel blade, cut off the posterior parts of the brain, the midbrain and the hindbrain, by cutting along the border of the cerebral cortex. Then cut off the olfactory bulb. Next, flip the remaining tissue so that the medial side is facing up. Now you need to remove the tissue covering the medial surface of the hippocampus. Using one spatula to hold down the tissue, insert the tip of the other spatula right below the corpus callosum and gently pull out the thalamus, the septum and underlying striatum and cut away this block of tissue. The hippocampus is now clearly visible with its banana shape. Using one spatula to hold the cerebral cortex down, place the tip of the other spatula under the ventral part of the hippocampus. Roll out the hippocampus and separate it from the cortex. Use caution as to not damage the hippocampus. Inspect the tissue for pieces of contaminating cortex. The cortex appears to be a different color from the hippocampus, so you can easily cut away any remaining pieces.